This session, we will discuss about the third classification of carbohydrate, which is polysaccharide. So under the basic classification of carbohydrate, we have monosaccharides, oligosaccharide, and polysaccharide. So already we have completed monosaccharide and oligosaccharide, in the disaccharide and trisaccharide also we have seen. And now we are going to see polysaccharide. So polysaccharides are the sugar polymers. So you will have more than 20 or more number of units of monosaccharide. So it can start from more than 20, sometimes hundreds, sometimes thousands of monosaccharides, which will be again joined with the help of glycosidic linkage. So you have to link with the help of, or you have to connect with the help of glycosidic linkage, which is again covalent linkage. So how we saw for the disaccharide and trisaccharide, it will be linked with the help of glycosidic linkage. The same way here also, thousands and thousands units of monosaccharide will be linked with the help of glycosidic linkage. Usually the polysaccharide is also called as glycanes. So most of the times the polysaccharide is called as glycanes, and here the polysaccharide units and length and type of linkage will vary from one polysaccharide to the other polysaccharide. Since you can have from 100 to 1000 or 80 or 20 above, based on the number of monosaccharide unit which is present, the molecular weight of each of the polysaccharide will vary. Usually your molecular weight will be depending on the number of monosaccharide units which is present, and the degree of branching, how much the branching of polysaccharide takes place, also depends on which kind of polysaccharide, where it is stored, how much amount of monosaccharide it gets to make polysaccharide. All those things are depending on so that the degree of branching will be accordingly. So this polysaccharide is further classified into homopolysaccharide and heteropolysaccharide. In today's class, we'll see only homopolysaccharides. So in the carbohydrate classification, we have monosaccharide, oligosaccharide, and then polysaccharide. Under polysaccharide, we have homopolysaccharides and heteropolysaccharides. So now we'll just see in this session only the homopolysaccharide. In the next session, we'll see heteropolysaccharide. In case of homopolysaccharide, you will have the same kind of monosaccharide units. So heteropolysaccharide means it will have different kinds of monosaccharide units, which we will be seeing in the next session. Homopolysaccharides will have the same sugar. For example, let's say if glucose is present in one of the monopolysaccharide in the sense, the same glucose unit will be present. It may be either 20 or 100 glucose will be there, or 1000 glucose molecule will be there. All will be interconnected with the help of glycosidic linkage itself. So most of the homopolysaccharide will be used for storage purpose. So it will be stored form of carbohydrate will be your homopolysaccharide. And here the monosaccharides, after the hydrolysis of this homopolysaccharides, will be used as fuels. So example, we have starch and glycogen. So starch as well as glycogen, both are homopolysaccharides, and it contains glucose as the monosaccharide unit. Starch, which will be obtained from plant source. So you can see the starch in potato. When you chop the potato, the white patchy layer which gets deposited in the water and which gets stuck to the surfaces and all is starch only. So starch is homopolysaccharide which is made up of glucose as the monosaccharide unit. And we have glycogen. Glycogen again is made of glucose units, but this glycogen is stored in the mammalian cell or animal system. So the storage form of polysaccharide is starch in plants. In case of animal cell, glycogen is the storage form of homopolysaccharide. So these two, both starch as well as glycogen, will be used for the fuel purpose, for the energy purpose. So both starch and glycogen will be oxidized to produce energy, and that energy will be used for the metabolic processes as well as the physiological processes. Apart from the starch and glycogen, which is a storage form of polysaccharide, you have other homopolysaccharides also, which is existing in the biological system, which includes cellulose, chitin, So the cellulose as well as this chitin, these are all the structural form of homopolysaccharides. So already what we saw is storage homopolysaccharide, which will be stored, and whenever we are in need of energy, those two will be taken, hydrolyzed, and it will be oxidized to produce energy. Whereas in case of this homopolysaccharides, especially in case of cellulose and chitin, these are the two structural elements which will be present in plant cell wall as well as in the animal exoskeleton. So whatever you could able to see on the surface of any insects or any animal tissue at all, it will be the thick layer of your chitin. Cellulose which will be present in plant cell walls. All the plant cell walls, grass, every plant, whatever we see, all the plants are made up of cellulose. And animals surface, whatever which is glassy surfaces we have in the insects and all, that is because of the presence of chitin. Chitin is one of the homopolysaccharide. So structural homopolysaccharide, which includes your cellulose and chitin. Storage form of homopolysaccharide, which includes starch and glycogen. So these are the homopolysaccharides which we have to consider of major importance in the biological system. So I'll just show the structure of homopolysaccharide, the general structure of homopolysaccharide. So your homopolysaccharide, which will have the same unit of monosaccharide. So I have shown with the same color. So you can see homopolysaccharide with same color. Glucose means all will be glucose. If it is galactose means all will be galactose. Then again here also glucose means glucose, galactose means galactose. So again this homopolysaccharide further can be classified based on how much, whether it is branched or not. So based on that, you have unbranched and branched homopolysaccharide. So you have branched and unbranched homopolysaccharides. So in case of unbranched homopolysaccharide, you will not be able to see any branched structure. You will have only the linear structure. There will not be any branches. Whereas in case of branched homopolysaccharide, you will have branches. Even in the branches, the same monosaccharide will be present. So the unbranched structure is also called as linear homopolysaccharide. So linear homopolysaccharide and branched homopolysaccharide will be classified. So most of the times you will have branched structure in case of the starch and in case of your glycogen. But when you do partial hydrolysis, we may get the linear structure, which you call it as unbranched structure. So what we should remember is the homopolysaccharide is also classified into linear homopolysaccharide and branched homopolysaccharide. So both here, whatever the linkage you are seeing, the linear one is also glycosidic linkage and the branch point also it is glycosidic linkage. Most of the times the linear structure is made up of alpha 1,4 linkage, and the branched structure, wherever you have a branching point, will have alpha 1,6 linkage. The rest of these structures, wherever I'm keeping cursor, are made of alpha 1,4 linkage itself. By this, we are completing homopolysaccharide. We'll see in the next session heteropolysaccharide and its importance. Thank you, students.